morning, students. Now we are going to talk about meiosis. So children, talking about meiosis, it is also called as reduction division because the production of offsprings by sexual reproduction involves the fusion of two gametes. So the process of meiosis, in which the production of offsprings occurs by sexual reproduction, which involves the fusion of two gametes. Children, in this case, the two gametes contains a complete set of haploid chromosome, and the gametes are formed from the specialized diploid cell. So this kind of cell division, which involves the reduction or which reduce the chromosome number to half, is called meiosis. Children, this special kind of cell division results to haploid daughter cells, reduces, that is why it is called reduction division, right? So we have discussed about meiosis, that it involves fusion of gamete, complete set of haploid chromosome we have discussed, and about the gametes, that is the specialized cell we have discussed. So actually the process of meiosis is basically in living organisms. It indicates the haploid phase in the life cycle of an organism, right? So whereas if we talk about the fertilization process, then it restores the diploid phase in the life cycle of an organism. Meiosis haploid gamete बनाते हैं, fertilization diploid zygote बनाते हैं, right? तो diploid से haploid और फिर haploid से diploid. Getting my point? So fertilization involved the diploid phase. So children, meiosis is generally include two sequential cycles in the life of an organism, which involve nuclear division and cell division, and which occurs by meiosis 1 and meiosis 2, right? So these are two sequential cycles which occur for the nuclear division and the cell division, and it involves a single cycle of DNA replication. So DNA replication have only single cycle. Now this meiosis 1, it initiate after the parental chromosome replicate to produce identical sister chromatid at the S phase. So meiosis 1 कब start होता है? ये initiate होता है जैसे ही parental chromosome replicate करते हैं and produces identical sister chromatids at the S phase. Then the process of meiosis, it involves the production of sister chromatids by the parental chromosome, and the process of meiosis also involves the pairing of homologous chromosomes and their recombination. And children, at the end of meiosis 2, 4 haploid cells are formed. So basically the meiosis 1 includes all the same processes as in the case of mitosis, right? And it includes prophase, metaphase, anaphase and telophase in both meiosis 1 and meiosis 2. So let us look at the meiosis 1. Children, because this reduces the number of chromosome from diploid to haploid, so meiosis is therefore known as heterotypic division, right? So let us discuss about the prophase of meiosis 1. The prophase of meiosis 1 is more elaborate and lasts longer than prophase of mitosis. It takes more than 90% of the time required for meiosis. Children, it may be divisible into 5 substages, that is leptotene, zygotene, pectine, diplotene and dikinesis. Talking about the leptotene, so in the leptotene stage, the chromosome condense and become visible. What happens actually, the centriole already duplicated and moves apart in pair. Astral rays appear around each of the centriole. Spindle begins to develop and chromosome differentiated from chromatin fiber. So they are double and each containing 2 identical chromatids. So compactation of chromosome occur, and this is followed by the second stage of prophase 1, that is with the zygotene. Now the next stage is the zygotene. During the zygotene stage, chromosomes start pairing together and this process of pairing of chromosome, that is the association of chromosome, is called synapsis. क्रोमोसोम की जो एसोसिएशन की जो प्रोसेस है ये सिनेप्सिस कहलाती है. सो जो पेयर बनते हैं क्रोमोसोम के, सच पेयर ऑफ क्रोमोसोम्स आर कॉल्ड एज होमोलोगस क्रोमोसोम. क्लियर? और फिर सिनेप्सिस के बाद जो क्रोमोसोम के टेट्रेड बनते हैं उन्हें बाइवेलेंट कहलाते हैं. So this synapsis is basically accompanied by the formation of a complex structure which is known as synaptomial complex. Children, however, this is more clear in the next stage of pectine. In the pectine stage, the pectine word comes from the another word which is pecus. Pecus means thick thread. So now during the pectine, the bivalent stage are clearly appear as the tetrad. Now what happens? They become thick and short and clearly visible as the diad. But actually they are the tetrad. As shown in this picture, they basically appear as the tetrad structure. ये इस तरीके का चार tetrad structure दिख रहा है, लेकिन actually they are the tetrad, but they look as the diad. एक पेयर ये और एक पेयर ये diad structure दिखता है. So in this homologous chromosomes, the two chromatid of the same chromosome is known as sister chromatid, right? And the two chromatid of different chromosome is known as non-sister chromatids. So the process of pectine involves recombination, that is the crossing over. And children, the recombination involves mutual exchange of corresponding segment of non-sister chromatid of homologous chromosome. So this process takes place by the breaking of segment, and that breaking is called nicking by the enzyme endonuclease, and the reunion is again there with annealing enzyme ligase, right? तो होता क्या है कि ये जो sister chromatids और नॉन सिस्टर क्रोमैटिड्स जो होते हैं, ये जो नॉन सिस्टर क्रोमैटिड्स होते हैं, इनके बीच में सेगमेंट्स का एक्सचेंज होता है. ये यहाँ से कट के दूसरे सेगमेंट में जाके मिल जाता है. So that recombination is being formed, and that process of recombination occur in that stage only. The fourth stage is diplotin stage. Diplotin is recognized by dissolution of synaptomial complexes. Children, homologous chromosomes which form bivalent separate from each other and accept the site of crossing over, right? So thus they form the X-shaped structure, right? So this X-shaped structure is known as chiasmata. Now the next stage is the dikinesis. It involves termination of the chiasmata. Children, bivalent chromosome align to the equatorial plate. The microtubule from the opposite pole of the spindle attach now to the pair of homologous chromosome. Right? Now the next step of meiosis 1 is the metaphase 1. The bivalent chromosome align on the equatorial plate, and the microtubule from the opposite pole of the spindle attached to the pair of homologous chromosome. So basically at the equatorial plate it is located then. Now the next step of meiosis 1 is known as anaphase 1. In anaphase 1, the homologous chromosome separates while sister chromatids remain associated at their centromere. Right? So centromere does not split. So chromosome number becomes half. Right? Next stage is the telophase 1. In telophase 1, the nuclear membrane and nucleolus reappear. Right? And it is followed by the cytokinesis. And this is called as diad of the cell. The stage between the two meiotic division is known as interkinesis. तो दो meiosis 1 और meiosis 2 के बीच की जो स्टेज होती है वो interkinesis कहलाती है. Right? Coming to the meiosis 2. Children, meiosis 2 is similarly like that of meiosis 1, having the same stages which are prophase 2, metaphase 2, anaphase 2 and telophase 2. And the meiosis 2 usually just after the process of cytokinosis. That is, before the chromosome undergo fully elongated, before that meiosis 2 initiated in the cell. When we talk about meiosis and compare the meiosis 2 with that of meiosis 1, we found that meiosis 2 is closely resembled to the normal mitosis. ये जो अगर हम कंपेयर करते हैं मियोसिस 1 और मियोसिस 2 को, तो ये समझ में आता है कि जो मियोसिस 2 है इट इज़ एग्जैक्टली लाइक द माइटोसिस. In the case of meiosis 2, the first phase, which is known as prophase 2, usually in this case the nuclear envelope disappear, जैसा कि हमने माइटोसिस में पढ़ा था. And as the time being, the chromosome get more compact. At the end of prophase 2, what happens? Nuclear envelope disappear and chromosome become more compact. तो यही सब चीज़ें हमने पढ़ी थी माइटोसिस में. The next stage is metaphase 2, in which the chromosome align themselves at the equatorial plate, and the microtubule come from the opposite pole of spindle and get attached to the sister chromatid at their kinetochore. Right? So metaphase में what happens? They get aligned at the equatorial, right? So that thing we have already discussed. And the microtubule from the opposite of spindle get attached to the sister chromatid by the kinetochore, right? Now the third step is the anaphase 2. In anaphase 2 of mitotic division 2, what happens? The simultaneously splitting of centromere occur, जो mitosis में भी हुआ था. The centromere hold the two sister chromatid together, cause the splitting of centromere. Chromosome move to their respective poles, right? तो यहाँ पे जो है न्यूक्लियर डिवीजन स्टार्ट हो जाता है, जैसे कि माइटोसिस में भी हुआ था, right? Now at the telophase stage, which is the last stage of meiosis 2, similarly what happens? The nuclear envelope reappear. ये nuclear envelope जो है ये reappear होता है and divide themselves into two nucleus, and these two daughter nuclei get surrounded by the nuclear envelope. And after this, the cytokinesis again follows the process, and the formation of cytokinesis result in the formation of tetrad haploid cells, right? So finally we have 4 haploid daughter cells. So this is about the meiosis 2. Now let us discuss about the significance of meiosis. Children, specific chromosome number of each species is achieved across generation in sexually reproducing organism by meiosis. So the number is maintained. It also increases the genetic variability in the population of organism, because the two gametes from the two different parents are involved, so that is why it leads to the variation in population of organism. It results in reduction of chromosome number by half. So because it reduces the number of chromosome, that is why it basically helps in the maintaining of number of chromosome, right? Because it helps in the gamete formation. It helps in the process of gametogenesis, that is gamete formation. तो अगर number of chromosome reduce नहीं करेंगे तो कहीं ना कहीं number maintain नहीं रहेंगे chromosome के, right? So that basically creates the problem, right? So and it also helps in the variation of the organism species. And for example, there is the figure on your screen which shows the variation in human population. Today we have discussed about meiosis cell division. So I hope that you understood all the concepts, all the processes, all the steps of meiosis, right? So children, still you found any kind of difficulty, then please feel free to ask. You can call me anytime, right? Till then, have a nice day and thank you.